The cardiovascular system starts developing at the beginning of week 3 of development. At that point, the embryo is a flat little pancake made up of two layers: the epiblast on the dorsal or backside, and the hypoblast on the ventral or front side. A line called the primitive streak appears on the epiblast back of this two-layered creature. Cells migrate along the primitive streak during gastrulation, and this gives rise to a three-layered embryo pancake, with each layer containing germ cells that form organs and tissues of the body. The ventral or bottom germ layer is called the endoderm, the dorsal or top layer is called the ectoderm, and the layer in between these two is called the mesoderm, and the heart derives from a part of the mesoderm called the visceral mesoderm. Okay, so let's take a transparent look at the mesoderm of this 3-week-old creature. Mesoderm cells go through the primitive streak and make their way up to the embryo's head. And this forms an area that's called the primary heart field. And it's horseshoe-shaped and has two limbs, one on either side of the future brain. This region lies on a blanket of endoderm cells which secrete vascular endothelial growth factor, or VEGF for short. VEGF signals the cells in the limbs of the horseshoe to self-organize into two heart tubes. A primitive pericardial cavity also appears lateral to each heart tube. At its inferior end, each heart tube connects to a vitelline vein, which comes from an extra embryonic tissue called the yolk sac, and this is how blood gets to the heart tube. Blood exits each heart tube at its superior end through a dorsal aorta, which then continues down the embryo's back. During lateral folding, the flat embryo goes from a trilaminar disc to a more cylindrical shape. The lateral borders of the embryo reach out towards each other like a self-hug and meet anteriorly at the midline, forming a cylindrical shape. This process makes the two heart tubes fuse into one, forming a single primitive heart tube. The left and right vitelline veins also fuse to form the sinus venosus, which is the inflow tract of the heart tube. Similarly, the two aortae fuse to form the aortic sac, which is the outflow tract of the heart tube. The two pericardial cavities also unite around the heart tube and form the singular pericardial cavity. The heart tube stays attached to the back wall of the pericardial cavity by a sheet of mesoderm called the dorsal mesocardium. The heart tube itself has two layers, an endothelial lining on the inside, which turns into the endocardium, and cardiac myoblasts on the outside, that give rise to the myocardium. Some of the myocardial cells in the sinus venosus start to produce a rhythmic electrical discharge at this early stage, but the conduction system itself and working myocardium are still underdeveloped, and they can't quite contract in perfect sync yet, so we still can't hear that familiar lub-dub sound. During craniocaudal folding, the now cylindrical embryo curves down its length and forms more of a shrimp-like creature, and this process pushes the heart tube down towards the chest. By the beginning of week 4, the heart tube reaches the thorax, and blood can be seen going through the heart tube. The heart tube develops sections. First, there's the sinus venosus, which has a left and right sinus horn that bring blood in, and above that there's the primitive atrium, and then the primitive ventricle, which are separated from one another by the atrioventricular sulcus. The primitive atrium gives rise to the left and right atria, and the primitive ventricle forms the left ventricle. The primitive ventricle is separated from the next region, called the bulbus cordis, by the bulboventricular sulcus. The first part of the bulbus cordis forms the right ventricle, as well as the outflow tracts for both ventricles. Finally, at the top of the heart tube, there's the truncus arteriosus, which pumps blood through the aortic sac into an early version of the circulatory system made up of the aortic arches. Clearly, this organization of structures doesn't look like the adult heart. So during week 4, the heart tube undergoes looping, which is a way of saying that the tube gets longer, its walls become thicker, and sections of the heart move around so that they end up in their right place. The heart tube's held in place inside the pericardial cavity by blood vessels at both ends, so looping starts with the heart tube folding into a C shape. The truncus arteriosus and bulbus cordis move down and to the right to form the top portion of the C, while the primitive ventricle bends to the right of the midline and a little to the front, forming the middle of the C. Finally, the primitive atrium and sinus venosus form the bottom of the C, and snuggle deeper inside the pericardial cavity. As development continues, the growing ventricle moves to the left, so it crosses over the midline again, covering the primitive atrium. By the end of week 4, the cardiac loop starts to take on the general appearance of the adult heart. Also, by this point, visceral pericardium attaches to the outside of the heart and forms the epicardium. Even though it kind of looks like a heart on the inside the definitive cardiac chambers have yet to partition. If we take a slice of the heart and look at it from the right side, we can see that the primitive atria and the primitive ventricles initially communicate through the atrioventricular canal. On the anterior and posterior walls of the atrioventricular canal, the mesoderm proliferates and forms an anterior and posterior endocardial cushion. These cushions grow toward each other and fuse. If we go back to the plane looking straight at the heart, we see that this separates the heart into left and right atrioventricular canals. On the ventricular side of both the right and left atrioventricular canals, endocardial cells proliferate to form the leaflets of the mitral and tricuspid valves, as well as the rest of the valvular apparatus. And this divides the atria from the ventricles. In the atrium, a tissue called the septum primum between the left and right atria grows downward, slowly creating two separate chambers by closing a gap called the ostium primum, which means the first opening. This septum primum then fuses with the endocardial cushion and closes the gap completely. Meanwhile, a hole appears in the upper area, called the ostium secundum, or second opening. Now we also have the septum secundum, which grows downward just to the right of the septum primum, and covers the ostium secundum like a curtain, leaving a small opening called the foramen ovale, and essentially creating a makeshift valve that allows blood to go from the right atrium to left atrium during fetal life. The ventricles separate when a muscular ridge of tissue grows upward from the apex, or the tip, and then fuses with a thinner membranous region coming down from the endocardial cushions. And there you have it, two separate atria and two separate ventricular chambers. Now, if we jump back to the outside of the heart, we see that each of these chambers is still connected through their outflow tract to the truncus arteriosus, meaning that blood just mixes from the two chambers. The truncus arteriosus starts to divide into the aorta and the pulmonary artery when two endocardial cushions appear on the right superior and left inferior walls. These grow with a spiraling trajectory, kind of like a corkscrew, and they wrap around each other, forming the aortico-pulmonary septum, which divides the truncus into the roots of the aorta and the pulmonary artery. So now blood gets routed to the right place. And finally, the semilunar valves develop shortly after. The rest of the aorta and the pulmonary artery come from the aortic arches. The aortic arches only exist briefly between weeks 4 through 6, and they sprout one after another from the aortic sac. There are 5 aortic arches on each side of the sac, and they're numbered from 1 to 6, because the 5th arch regresses and doesn't turn into any adult structures. The 1st and 2nd arch disappear pretty quickly too, but they leave behind the maxillary and the stapedial arteries, respectively, coming off the internal carotid arteries. The 3rd arch gives rise to the 2 common carotid arteries, as well as part of the internal carotid arteries. The left 4th arch forms the aortic arch, and the right 4th arch forms the right subclavian artery. The 6th arch forms the pulmonary arteries, and on the left side, it also forms the ductus arteriosus, which is a fetal structure through which blood from the pulmonary artery shunts directly into the aorta, which bypasses the immature lungs. Now, rewinding just a tad and looking at our primitive inside of the heart again, we can't forget about the other venous side of the heart, or the sinus venosus, which initially opens in the center of the primitive atrium. Over time, the sinus venosus becomes asymmetric. It shifts over to the right, with the right sinus horn becoming the smooth-walled part of the right atrium, as well as the openings for the superior and inferior vena cavas. Meanwhile, the left sinus horn shrinks and persists only as the coronary sinus and the oblique vein of the left atrium. In the wall of the sinus venosus, a special group of myocardial cells organize and synchronize their electrical discharge, forming the sinoatrial node and taking over as the cardiac pacemaker near the entrance of the superior vena cava. Soon after the sinoatrial node is developed, another group of cells found in the atrioventricular septum organizes into the atrioventricular node, which is the secondary pacemaker center. At the same time, cells in the interventricular septum differentiate into specialized conducting cells that form the bundle of His. Cells in the rest of the ventricular myocardium differentiate into conducting cells that form the Purkinje fibers. Alright, as a quick recap, the cardiovascular system starts developing from the primary heart field region of the mesoderm during week 3 of prenatal life. It starts as a horseshoe-shaped structure with a pair of tubes that fold and loop around to put the primitive heart structures into the right positions. Afterwards, cardiac septa develop and partition the heart, giving rise to 2 ventricles and 2 atria. The conduction system of the heart's initially in the sinus venosus, but when the sinus is absorbed by the right atrium, pacemaker cells become the sinoatrial node in the wall of the right atrium.